on what you can observe and classify organisms into the same group that traditional approach. Today, we apply molecular approach, molecular sequence data. The data we generate DNA sequence, protein sequence, and based on the sequence, we can construct relationship between organisms. That's the modern approach, molecular approach. For traditional approach, any limitation from traditional approach, because some organisms they have similar characteristics, but they don't have relationship. Okay, even similar, but they don't have any relationship. That are similar to the plant. They have similar characteristics, but they don't have any relationship. That are limitation from the traditional approach. For molecular approach, more advanced, we can compare between organisms even they are very distant. Have relationship between them. We measure it. Okay, even distant, for example, we can compare human genome with banana genome. Animal and plants, we can compare. Because we based on DNA or protein. If you based on morphology, you cannot compare animal with the plants. That's the advantage when you apply molecular approach. This example show you phylogenetic traits. And we use probing. For this case, we say the genes in proteins, and we base on protein. And protein isolate from different organisms. So that's why you see the name of organism. But here we don't base on morphology. We base on protein and protein probing. You already studied in lecture 5, alignment. The definition of autolog. Autolog that means protein present in different species, but they have common ancestor. That means protein here homologous sequence. Otherwise, we cannot compare because they have common ancestor. Before we go detail with the structure of the phylogenetic trees, we look at the definition. The definition: phylogeny. Phylogeny, evolutionary histories and relationship of organism, gene or protein. So that means phylogeny show you what? Phylogeny show you relationship between organism. Relationship, how close between them, how distant between them. That's the relationship. The second one also histories. So that means show you when the events happen. When the event happens. Million years, 700 million years ago, 500 years, a million years ago, that's when the event happened. Event here, one common ancestor split into different species, new species when it happened at the evolutionary history. Gene or protein. So relationship between gene or protein, but actually gene or protein and animation need to isolate from one species, So this one is protein and isolate from species. Okay, so that's why the tree will show the name of the species. Phylogenetics. Phylogenetics is the study of evolutionary histories and relationship of organisms, gene or protein. The phylogeny and phylogenetics, that means the field of study phylogeny. Phylogenetics also provide a basis for comparative genomics. Comparative genomics. Let's introduce the term genomics. That's right. Transcriptomics, proteomics. Okay. For genomics, we classify into structural genomics, functional genomics, and comparative genomics. Structural genomics, that means identifies the gene structure, where the gene locates on one chromosome and how many genes on one chromosome. That is structural genomics. Functional genomics study function of gene or protein, because protein is the product of gene. Okay? Function of gene or protein, that means transcriptomics and proteomics. Okay, transcriptomics and proteomics belong to functional genomics. And the last one you look at today, I just introduce you already, we don't study detail, comparative genomics, study of interrelationship of genome of different species. We can compare the whole genome from one species with the other species. We compare the whole genome. For phylogeny, we become specific gene or protein, just single. But comparative genome, we compare the whole genome. And when compare the whole genome, we can identify region of similarity and different among genome. Phylogenetic traits. We view the traits, phylogenetic traits, or evolutionary traits. Diagrammatic representation of evolutionary relationship among various taxa in the protein. So the tree actually diagram. In diagram here represent relationship between organisms. Represent relationship. So that means how relationship between horse and dog and kangaroo compare with the other one. When we move further with the phylogenetic tree structure, we'll explain detail the tip, the branching. Okay, so relationship between represent evolution relationship. Can construct with morphological data, as I mentioned, based on morphology, what we observe, and molecular data. Today we study molecular data, that means DNA sequence and protein sequence. A hypothesis, not a reality. Hypothesis, that means we predict the event happen and how long ago it happened, how relationship between them. We base on DNA or protein sequence, not reality. Because we cannot check the box, because the box already over, okay, million years ago. Reflects our understanding at a given time. Before, we don't have molecular approach. We cannot say we have a relationship with banana. Okay, that reflects our understanding. Now, with molecular approach, we can show the evidence how much relationship between us with banana, how much gene, percentage of gene we share with banana. We move detail with the phylogenetic tree structure. Two things. The first one we call components, and the second one topology. Not. Look at not. We have the circle here. The circle we call not. Okay? Not the circle, and we classify into at the end of the branch, the tip of the branch, we call terminal knot. And where two branches join together, we call internal knot. So what is the difference between terminal and internal? Terminal knot, operational taxonomic unit, also have the other name. Operational taxonomic unit, that means represents for species or gene or protein. So that means here represent for species you compare T1, 2, 3, 4, 5. That means human, chickens, mouse, that the species you compare. Terminal. Internal, not hypothetical taxonomic unit, hypothetical taxonomic unit represent the last common ancestor. Okay? Where here we call internal, and internal represent common ancestor. From common ancestor, new species, split into new species T2 and T3. That's the common ancestor. Common ancestor, hypothetical. Hypothetical, what it means? Because we cannot identify specific species here. For example, T2 human, T3 mouse, we cannot identify what is common ancestor. Monkey or fish, we cannot do it. Just common ancestor. Identify common ancestor. That means share, high similarity, and then identify common ancestor. Just that all. You cannot identify is that specific species. Which species common ancestor? We cannot do it. So that's why hypothetical taxonomic unit, internal not represent for common ancestor. And now look at relationship between organisms with the common ancestor. We have the sister group and our group. Descendants, T2 and T3, we call descendant. Descendant that means compare with common ancestor here. Okay, T2 and T3 common ancestor have the same common ancestor, and we call descendant. Descendant that means compare with the common ancestor. And T2, T3 share the same common ancestor, we call sister group. T1 not share this common ancestor with T2 and T3, we call our group. We got not the same common ancestor, but depend where you look at. Polytry show you many internal not represent for common ancestor. 1, 2, 3, 4. If you look at this common ancestor, only 2 species in sister group T2 and T3. If you look at this common ancestor, only T4 and T5. But here, you look at this common ancestor, because where the branch join together, that means we call common ancestor. And this common ancestor have how many species? 4. T2, 3, 4, 5. If you look at this common ancestor, the common ancestor for all species, okay? So we have all species with this common ancestor, the root. Okay, so depend where you look at the common ancestor. And then the number of species present in that group different, not the same. Okay? Later, we have the term play, we explain more detail. So here, to explain about the phylogenetic tree structures. So the first one, component, the first component, not the first component, and different term used. So I explain to you, understand why we use different term here. Range, the second one, range represent the time estimated of the evolutionary relationship among the taxonomic unit. Okay, based on the branch long or short represent how long ago. Okay, 500 million years ago, 100 million years ago. So the time, that means the branch will represent for the time, how long ago. And based on what we can, the scientists can estimate how long ago. For example, we compare between two sequences, and between sequence look at A1, the same, A1, A5, okay? One amino acids, one amino acid different for the horn, for the horns. And